applied plant pathology or chemicals in plant disease management. So now we'll see the probability topics or the probability questions that we can expect in ASRB Net 2023 plant pathology. So my name is Shankar Reddy and I'm currently working as assistant professor in Kasar Best IU. So coming to the classification of fungicides, in classification of fungicide, there are many classifications are there. Let me tell you some important concept here. Fungicides are classified based on their chemical nature, like if their composition is composed of major composition is composed of copper, it is called as copper fungicide. It is a sulfur fungicide, or mercury fungicide, something like that. Their major composition, based on their major composition, they are divided. So let's discuss about copper fungicides. It's very important. Copper fungicides in plant disease management. First one is Bodo mixture, otherwise called as Bodax mixture. It was first discovered, or sorry, first developed by, or first discovered by PMA Milladet in 1882 to 1885. So this Bodo mixture is developed by Milladet for the management of grapes downy mildew. So grapes downy mildew is caused by Plasma para viticola. So in that time in 1878, grapes downy mildew is a very serious disease in grape wine production, especially in France. So where Milladet came up with this Bodax mixture. It is also called as Bodo mixture. It is also called as holy water. It is also called as holy water or miraculous fungicide. It's been almost 130 years, 150 years since the discovery. Still the farmers are using this Bodo mixture for the management of various kinds of plant diseases. So let me tell you, and it is also called as serendipity discovery. Serendipity means accidental discovery, where Milladet discovered accidentally. So the composition is 1 kilogram of copper sulfate, 1 kilogram of hydrated lime or 100 liters of water. This can be mixed and can be applied on the crops. And if you are preparing this, we need to apply on crops within 1 day, because if it goes more than 1 day, there might be a toxicity effect or something like that. So the composition is 1 kilogram of copper sulfate, 1 kilogram of hydrated lime, 100 liters of water. That is 1:1:100. And this Bodo mixture can be recommended for downy mildew of grapes, blights, anthracnose. And the next one is Bodo paste. So instead of 100 liters in Bodo mixture, we are using just 10 liters in Bodo paste, because we need to make it into paste, right? So that's why we are using 10 liters of water. That is 1:1:100. 1 kilogram of copper sulfate, 1 kilogram of lime, and 10 liters of water. So this can be recommended applying on wound areas, especially citrus gummosis and coconut stem bleeding, to prevent the further invasion of invading of pathogens. The second one is burgundy mixture. Burgundy mixture, otherwise called as soda borax. Soda borax. This question was asked in 2019. So which of the following fungicide is called as soda borax, or the composition of soda borax, because burgundy mixture is otherwise called as soda borax, right? So it is also called as Mason lime, because the scientist who discovered it is Mason in 1887. This question was asked in 2021 net. Burgundy mixture was developed by Mason 1887. Okay, burgundy mixture is otherwise called as soda borax, otherwise called as Mason lime. Okay, it was developed by Mason in 1887. The composition is 1 kilogram of copper sulfate, 1 kilogram of sodium carbonate, and 100 liters of water. That is 1:1:100. So we can use stem bleeding or coconut stem bleeding disease, or that is cholera paradoxa, which is caused by. So this disease we can use burgundy mixture. And chestnut compound, which was developed by Beverly in 1921. So the composition is 2 parts of copper sulfate, 11 parts of ammonium carbonate. So this can be recommended for the damping of disease, that is pythium damping up. The next one is Chaubatia paste, which was developed by Singh in 1942. So the composition is 800 grams of copper carbonate, 800 grams of red lead, and 1 liter of linseed oil or linoleic oil. The composition is 800 gram, 1 liter. And we can use this Chaubatia paste against pink disease of rubber or stem canker or apple and pear, something like that. So this is a very important. We may expect 1 or 2. Bodo mixture was developed by Milladet 1882 or 1885. The composition is 1:1:100, copper sulfate, lime, and water. So burgundy mixture, sorry, Bodo paste, instead of 100 liters of water in Bodo mixture, we are using just 10 liters to make it as a paste. Burgundy mixture, the main difference between Bodo mixture and burgundy mixture is, in Bodo mixture lime is used. In burgundy mixture, instead of lime, we are using sodium carbonate here. We are using sodium carbonate. If it is a lime, it is Bodo mixture. Instead of lime, if you are using sodium carbonate, it is a Burgundy mixture that was developed by Mason 1887. Chestnut compound Bailey 1921, 2 parts of copper sulfate and 11 parts of ammonium carbonate. And Chaubetia paste 800 grams of copper carbonate, 800 grams of red lead, and 1 liter of linseed oil, linolenic oil. So the next one is mode of action of some important systemic fungicides. So in every year we can expect 1 or 2 questions from this mode of action side. And I have given some important. I had calculated, or I had gathered some important fungicides and their mode of actions. And azoxystrobin and trifloxystrobin mode of action is mitochondrial electron transport inhibitor. Benomyl, that is mitosis and cell division inhibitor. Benzimidazole inhibits beta tubulin biosynthesis, very important. Benzimidazole group of, so mode of action of benzimidazole, that is inhibits beta tubulin biosynthesis. Captan is otherwise called as Kittelsen killer, because the fungicide was developed by or discovered by Kittelsen. That's why it's called as Kittelsen killer. The year of discovery is 1952. It's a very, very important year also, that was asked previously. So Captan otherwise called as Kittelsen killer. It is a seed treatment fungicide, majorly recommended for seed treatment, and inactivation of enzymes through interaction with their NH2 and SH group. Carbendazim, benomyl and thiobendazole, chitin and nucleic acid biosynthesis and mitosis and cell division inhibitors. Carboxyn and oxycarboxyn, these two are the first systemic fungicide. It is a very, very, very important. Last year also it was asked, what is the first systemic fungicide that is developed? Carboxyn and oxycarboxyn. The carboxyn and oxycarboxyn belonging to group oxanthin group of fungicide. These two are the first systemic fungicide to be discovered by Van Schemling and Marshall Kulka. Van Schemling and Marshall Kulka in 1966. They developed or discovered first systemic fungicide, that is carboxyn and oxycarboxyn, or we can simply call it as oxanthin group of fungicide. Mode of action is interfere with electron transport chain. Chloramphenicol inhibits 15S ribosome function and inhibits translation step of ribosome function. Cyclohexamide inhibits protein synthesis. Edifenphos and iprobenphos, phospholipid biosynthesis. Erythromycin inhibits 15S ribosome function. Phosphatyl aluminum, inosine and ketogen inhibits spore formation and penetration of pathogen. Hexagonazole, sterile biosynthesis inhibitor. Iprodine, lipid and membrane biosynthesis inhibitor. Mancozeb, that is enzyme and protein degeneration. Metaloxyl, very, very important. Metaloxyl, it disrupts fungal nucleic acid biosynthesis. So this question we can also expect, which of the fungicide having both acropetal and basipetal action? The first one is phosphatyl aluminum and the next one is metaloxyl. If the option was not there, phosphatyl aluminum, we can go with metaloxyl, because this question was asked last time 2021. So which of the following fungicide having both acropetal and basipetal, are like both upward and downward direction movement are there mode of in their action? So the first one is phosphatyl aluminum, and the next one is metaloxyl. If there is no phosphatyl aluminum, we can go with the metaloxyl. Okay, their mode of action is fungal nucleic acid RNA synthesis disruption. And penicillin inhibits the synthesis of murin or murin biosynthesis. Polymyxin B destroys cytoplasmic membrane. Streptomycin inhibits 13 ribosome function. Tetracycline inhibits binding of amino acid tRNA with ribosomes. Thiophenate or thiophenate methyl, mitosis and cell division inhibitor. Triazole, sterol biosynthesis inhibitor. Tricyclazole, melanin biosynthesis, it is very, very important. The mode of action of tricyclazole, it is a melanin biosynthesis inhibitor, or inhibits the melanin with the aprosorium, within the aprosorium, sorry. So this tricyclazole is the specific fungicide recommended for the management of rice blast. What is the specific fungicide recommended for rice blast? So tricyclazole is the specific fungicide recommended for rice blast. Not only tricyclazole, this edifenphos and iprobenphos is also recommended for the management of rice blast. So rice blast, what is the specific fungicide recommended for rice blast? Tricyclazole. B, edifenphos or iprobenphos. D, all the above, something like that they will ask. But specific fungicide is tricyclazole, but edifenphos and iprobenphos is also recommended for the management of rice blast. And the trade name of tricyclazole is blastin, or blast or blastin, something like that. So tridomorph, which is a sterol biosynthesis inhibitor. This tridomorph is called as rescue treatment fungicide, that I will tell you in the later slides. Mycoherbicides. So we are seeing some mycoherbicides, which means herbicide which are actually isolated from the fungus, and they are using it as a herbicides. So their chief source is isolated from fungus, and they are exploiting against weeds. Okay, now we'll see coligo. Coligo is a one herbicide which is isolated from a fungus called Coltrotrichum griosporides, Pharma species and it is actually targeted against echnomine, targeted against like a giant witch weed. The scientific name of giant witchweed is echnomine, same, the echnomine virginia, and the crops are rice and sorghum. The next one is divine. It is a mycoherbicide isolated from Phytophthora palmivora and exploited against milkweed, vineweed, that is a Moronia odorata, and citrus orchids, and cassette, cassette, which is actually isolated from Alternaria cassia, and Jimsonweed and sickle pod can be controlled by using this mycoherbicide. The scientific name is Datura stramonium, and soybean and peanut. Biomal, Coltrotrichum gliosporides, Pharma species malvae, and mallow round leaf or mallow weed, that is a dwarf mallow or round mallow, which is caused by Malva pusilla. Wheat, flax, and lentil. Velgo, isolated from Coltrotrichum cocoides, velvet leaf, and the scientific name is Abutilon theoprasti, soybean, and Lubova number 1 and number 2 isolated from Coltrotrichum gliosporides, Pharma species cascute, and Coltrotrichum gliosporides, that can be controlled by using exploited against dot run as well as silkachia, Cascuta sinensis and Cascuta australia and Heckia seria, and serike, and soybean crop. Stump out, that is Cylindrobasidium leve, Australian wattle, and Acacia miransi, and Acacia pisantha, and native vegetation, and Biocon, Conodendron purpureum, Biosid, Puccinia canaculata, and there are all the various kinds of mycoherbicide that is exploited against weeds. So this is a very important, like I have seen like Coligo, Divine, Cassette, Biomal, Velgo. These are all that was asked before in previous questions, like it is actually isolated from, or maybe what is the target weed, or what is the scientific name of the target weed, something like that they will definitely ask. So the next one is the disease is actually believed to be introduced from. So actually disease to be introduced from. The first one is coffee rust. So coffee rust is caused by Emilia vastatrix. That is, this is believed to be introduced from Sri Lanka. In 1866 it was recorded in Sumatra. 1878 it was recorded in Java. 1889 it was recorded in Philippines. Okay, and lead blight of potato, Phytophthora infestans, actually introduced from Europe. And the rest of Chrysanthemum Puccinia horina introduced from Japan and Europe in 1904. Flax smut of wheat is very important. Flax smut of wheat is caused by Eurocystis triticea. It is actually introduced into introduced in India in 1906 from Australia. So these are all diseases are believed to be introduced from other countries to India. Okay, in India the flax smut of wheat was first reported in 1906 from Australia. Downy mildew of grapes, Plasmapara viticola 1910 Europe. Powdery mildew of crucifer, Podospora xanthi 1910 Sri Lanka. Downy mildew of cucurbits, Pseudospanospora cubensis, but was reported first time in India in 1918 from Sri Lanka, introduced from Sri Lanka. Downy mildew of maize, it is believed to be introduced from Java. And paddy blast, Magnoportha graciae, it is believed to be introduced from Southeast Asia. As I told you that in previous presentations, in 1908, rice blast was first reported in India in the year of 1918 and 1919 time, especially in Tanjore district of Tamil Nadu. The next one is black rot of crucifers, Xanthomonas campestris, Pathovore campestris. So this Xanthomonas campestris and Pathovore campestris, that is black rot of crucifers, enters into the plant cells through hide thorns. It's a very important. And introduced in India 1929 from Java. And I have given like this. I have given some. So please. You guys please go through. And powdery mildew of rubber introduced from Malaysia. Black shank of tobacco introduced from Holland. Fire blight of apple and pear introduced from England. But what I was a little bit doubted about, in many books I have seen that fire blight was not reported in India. But here you see the first record was already reported in 1940 itself. Then how can we say that the disease was not reported in India? But I have no idea what exactly they are trying to tell. But I know what is my experience or my knowledge is that I have studied so many articles and all those things that fire blight of apple was already reported in India. Okay, please let me come to know if I'm wrong. So banana bunchy top virus introduced, believed to be introduced from Sri Lanka. Crown gall of apple and pear introduced to from England. Potato wart introduced from Netherlands. Onion smut introduced from Europe, and golden nematode of potato introduced from Philippines. Bacterial blight of pad introduced from Philippines, and golden nematode of potato introduced from Europe. Downy mildew of onion introduced from Europe. Downy mildew of sunflower introduced from Australia. So this is important. In the last year, this question was asked. Downy mildew of sunflower pathogen, or downy mildew of sunflower is caused by Plasma para halsteadi. Plasma para halsteadi. Okay. And peanut stripe virus, which is believed to be introduced from China in 1987. So these are all the various important diseases that is believed to be introduced from, and introduction year was given here. Hope we can expect maybe 1 or 2 questions from this. So the next one is the list of pests and diseases under domestic quarantine. Under, you know very well, there are 3 types of quarantine: international quarantine, embargo, and domestic quarantine. So now let's discuss about domestic quarantine, some important pests and diseases of domestic importance in India. So the questions will ask like this. I will tell you: San Jones scale, that is Quadrispidiotus perniciosa, which was noted in 1953. Fluted scale, that is Isaria purchasei, and potato cyst nematode Globodera rostrosceniensis and Globodera pallida. Codling moth, that is Carpocapsa pomenella, and coffee berry borer, and banana bunchy top virus, banana mosaic virus, potato wart, apple scab. These are all under domestic quarantine. Domestic quarantine means, imagine if it is infected in the particular state, if it is a very dangerous disease, so the transportation of the particular material from one state to another state is completely prohibited under domestic quarantine. So the examples will be asked like this in exam. So which of the following diseases under domestic quarantine in India? A. Potato wart. C. Banana bunchy top. D. Potato wart. Apple scab. Something like that they will ask. So these are all are under domestic quarantine. So San Jones, that is Quadrispidiotus perniciosa, fluted scale, potato cyst nematode, banana bunchy top virus, potato wart, and apple scab. These all are under domestic quarantine, and these materials cannot be transported from infected state to other states of the India. The next one is classification of fungicides based on their chemical nature and mode of action. So here actually divided into 2 ways, that is systemic and non-systemic. So let's discuss about non-systemic. So copper, as I told you, we already discussed in the first slide itself, that is Bordeaux mixture, Burgundy mixture, chestnut compounds, and their mode of action has been given here along with what is the targeted pathogens. So sulfur, so wettable sulfur, elemental sulfur, lime sulfur, these are all comes under the fungicide group sulfur. Mercury fungicide, mercury chloride comes under mercury. Dithiocarbamates like mancozeb, thiram, and thiram, ginab, nabum, these all are also used as a seed treatment fungicide. We can expect questions like this. See, dithio, which of the following group of fungicide, or which are following fungicide group can be especially exploited as a seed treatment? So if you see thiram, zinab, nabum, mancozeb, they all are especially exploited against seed treatment. So we can expect such kind of questions. Heterocyclic nitrogen compounds, that is captan. Captan is otherwise called as Kittelson killer because it was discovered by a scientist called Kittelson in the year of 1952. The next one is the phosphate. The fungicide comes under phosphate is phosphatal aluminum. As I told you that it is the best example of fungicide with both acropetal and basipetal, or we can also call it sympatal. Okay, and guanides, that is dodine. This is about systemic fungicide. Carboxin, as I told you, the first systemic fungicide to be discovered is carboxin. So that is the oxathin group, oxathin group or carbamidine groups. So the fungicide first discovered systemic fungicide is oxycarboxin and carboxin. The trade name of oxycarboxin is plant wax, whereas the trade name of carboxin is Vitavax. So this oxycarboxin is especially recommended exploited against rust diseases, whereas this carboxin is especially exploited against smut diseases. So first systemic fungicide, systemic fungicide was discovered by Van Schemling and Marshall Kulka, that is oxycarboxin and carboxin, and discovered by Van Schemling and Marshall Kulka. Year is also very important, 1966. and dinitrophenols, dinocap, strobilurin. These are all the new generation fungicide. Are actually these fungicides are isolated from a fungus called, or mushroom. We can say mushroom. Both are same, fungus or mushroom. These are actually strobilurin group of fungicides, are new generation fungicides. They're actually isolated from a mushroom called Strobilurus tenacellus. Strobilura tenacellus, it is also very important. And the strobilurin group of fungicides are azoxystrobin, picoxystrobin, trifloxystrobin. These are all the different kinds of systemic fungicide that comes under strobilurin group. Then phenylamide, phenylamide, phenylamides, that is metaloxyl and binoloxyl. These are all the phenylamides, and pyrimidinols and erythrimol and dimethrimol, and benzimidazoles, very very important. This is also benyl carbendazim, thiophenate methyl sir comes under this benzimidazole. Then thiodiazoles and erythrodiazole, that is comes under this thiazolol group, and organophosphates, edifenphos, iprobenphos, and pyrazophos. As I told you that not only tricyclozole, this edifenphos and iprobenphos are also exploited against rice blast disease management. Then anilopyrimidines, cyprodenil and mepanixim and pyrimethanol. These are all the some fungicide that comes under this group. And MBI, that is carboxamide and cyclopropane. And antibiotics like streptomycin, blastcytine, causagamycin, validamycin, these are all comes under this antibiotics groups. And their mode of actions has been given on the right side. You guys just please go through, because because we already discussed this mode of action, all those things. Wherever the fungicide, whatever I mentioned, is more or less similar here. So other topics that we can expect is like special methods of application of fungicide, like a trunk injection, or like sucker treatment, or like trunk injection, sucker treatment, or maybe pairing and preliminage, capsule application. These are all some kind of treatments. And the treatments or techniques that is recommended for what kind of disease. For example, if it is a trunk injection, we can recommend it for coconut budrot, that is coconut, we can also call it budrot, right? So such kind of diseases, and capsule injection, all those things, that is also an important. And antifungal and antibacterial antibiotics in plant disease management. For example, I will tell you one thing. So the best example of antifungal antibiotic is Ariofungin. Ariofungin is the best example of antifungal antibiotic that was first developed by M.J. Tirumalachar. He is also considered as father of plant disease father of antibiotics in India. So the first antifungal antibiotic is Ariofungin, that was developed by M.J. Tirumalachar. M.J. Tirumalachar. Not only this Ariofungin, like dermostatin, MYC4, so many other fungicides, antibiotics, and bacterial and fungal antibiotics were developed by this M.J. Tirumalachar. And about quarantine, and maybe some important things about quarantine also, we can expect some questions. For more information, you guys can mail us at or you guys can reach us at the description below. Stay geeky, stay tuned. We are Team Geeky Researchers.